When many people think of E. coli, they envision a sickening, invasive, and harmful bacteria which can be contracted by improper handling of food or consumption of undercooked meat. Of course, they also envision the intestinal issues which accompany its consumption. However, scientific fact proves that there are many bacteria, including certain harmless strains of E. coli, which are part of the normal flora of the human body. In the mid-2000s, microbiologists began to chart the vast numbers of organisms that live within the human body. This diverse collection of organisms is termed the microbiome. Over time, scientists called synthetic microbiologists began to experiment with innovative ways to tweak the microbiome's genes to aid humans. These dedicated microbiologists built on the fact that genes typically do not work in isolation. In fact, many genes communicate with each other to activate and deactivate each other's activity. This experimentation took advantage of this fact and manipulate these communications by creating new cells from existing ones. The newly created cells would respond to signals in new ways. Initially, this breakthrough technology was thought to be used in the outside world as environmental sensors, perhaps to detect airborne pathogens or biological weapons. However, microbiologists such as Dr. James L. Collins and Timothy K. Liu are discovering ways to utilize this technology in the human body and search diseases to utilize the tweaked microbiome. Collins and Liu co-founded a company called Synlogic in 2013. Their goal was to search out diseases that microbiome technology could prove useful. One of the top picks is a rare inherited disease called phenylketonuria, or PKU for short. Victims of PKU must avoid dietary protein found in most meats and cheeses because their bodies cannot break down an amino acid byproduct of the protein called phenylalanine. As phenylalanine builds up in the body, it has a potential to damage neurons in the brain. This damage may lead to delayed development, intellectual disabilities, and psychiatric disorders. In a response to promoting a more effective treatment of PKU, Dr. Collins and colleagues enlisted the help of genetically engineered E. coli. The researchers inserted genes into the E. coli bacteria's DNA so that once inside a person's gut, it would break down phenylalanine in a similar way to a healthy person's cells. To date, the newly encoded E. coli bacteria have proven successful in trials with first mice, then monkeys, and lastly healthy human volunteers. Because of the proven success with test volunteers, the altered bacteria are now staged to be tested on people infected with PKU. Synlogic's PKU treatment is paving the way to become the first synthetic biology medical treatment to gain approval by the FDA. A former student of Dr. Collins, Dr. Jeff Hasty, who now runs a microbiology lab at the University of California, San Diego, makes note, "You can't overestimate the impact of the microbiome work." He further explains that, in a nutshell, microbiome discoveries change everything. In fact, in July, Dr. Hasty, along with fellow colleague Dr. Danino, published a review in the journal Cell Systems. The review catalogs many other disorders researchers are designing synthetic microbes to treat. These diseases include inflammation and infections, as well as cancers.